The quest to conquer pain without, that shadow of addiction has really driven scientists for generations.
Absolutely.
And you think about those early chemists facing the incredibly complex structure of morphine. I mean, it was like a puzzle box, wasn't it? So intricate.
Yeah, 1,000 tiny interlocking pieces.
Yeah.
They were trying to understand its secrets, figure out how to build it themselves, basically.
Right.
And that complexity, that inherent structural puzzle, that's really where our exploration starts today.
Okay.
We're doing a deep dive into the chemistry of opioids, drawing insights from this collection, Chemistry of Opioids, Volume 299. It's from 2011, edited by Hiroshi Nagase.
A pretty comprehensive source, then.
Oh, yeah. Think of it as your shortcut to understanding a really huge field.
Nice. So this isn't just scratching the surface.
No. This volume is packed. I mean, cutting-edge research from the basics of synthesizing morphine and its chemical cousins,
Right.
all the way to developing completely new opioid-like drugs, ones that target specific receptors in our bodies.
And it even gets into the clinical trials, seeing if these ideas actually work in people.
Exactly. We get glimpses of that, too.
So what's our mission for this deep dive? What are we trying to pull out of this?
Okay, so the mission is really to extract the key chemical strategies. What are researchers actually doing to develop pain meds that are hopefully more effective and less addictive?
And not just for pain, right? You mentioned other things.
Yeah, we'll also touch on treatments for things like persistent coughs and that really frustrating itch that can happen with certain conditions. It's a story full of, significant scientific hurdles and some really clever solutions.
All right, let's start with that first puzzle then. Morphine itself. You said it was complex. What made it such a beast for those early chemists?
Well, morphine has this core structure. It's called a 4,7,5 epoxymorphine in skeleton.
Okay.
Imagine several rings of atoms all fused together. But the really tricky part is it has 5 asymmetric centers, one right after the other.
5. Wow. Like specific connection points.
Exactly. Think of them like Lego bricks that only fit one way. Plus, you've got other bits. A phenolic hydroxyl group, that 4,5 epoxy ring, which is an oxygen bridging 2 carbons, and a basic nitrogen atom, all positioned just so. It's this 3D complexity that made building it from scratch a massive hurdle.
So it wasn't just connecting the dots. It was getting the exact 3D shape.
Precisely.
So instead of trying to copy nature perfectly right off the bat, did they try to make simpler versions, things that might be easier to produce?
That was definitely part of the strategy early on, design simpler, related molecules that still had pain relief but could be made industrially. Sort of a good enough for now approach.
Makes sense. Get something out there.
But morphine itself, its unique properties just kept scientists hooked. I mean, our source highlights that between 2004 and 2009 alone, 8 different research groups published work on the total synthesis of morphine and related compounds.
8 groups in just 5 years. That really shows how important cracking that code was.
It does. It shows the ongoing intellectual challenge and the fundamental importance of knowing how to build these molecules.
So were there common strategies? Any clever chemical tricks these groups were using?
Yeah, several key approaches popped up. For instance, Overman's group used something called an intramolecular Heck reaction.
Okay, what's that, basically?
Think of it as a way to snap together 2 parts of the same molecule to form a new bond, specifically creating one of the ring structures you see in morphine.
Building the rings internally.
Exactly. And Ogasawara's team used a different approach, a cyclization followed by dehydration, basically forming a ring and then removing a water molecule using an aldehyde as a key piece. Cheda later adapted that for a related molecule.
Interesting. Any specific examples that really stand out?
Well, the Cheda group's synthesis of codeine, which is very close to morphine, is a good one. And the Guo group's synthesis of racemic codeine, that's the mixture of mirror images.
Right.
Chera's synthesis used that Heck reaction we mentioned, but they had to carefully build a key intermediate first, getting the 3D shape just right. Guo's approach really focused on building one specific ring very selectively. It shows the level of control you need.
And you mentioned those asymmetric centers, the sort of handedness of the molecule.
Yeah.
How did they control that? Make sure they got the right version?
Yeah, that's crucial. It's called asymmetric reduction. You need the molecule to have the right handedness to fit its target in the body, like putting a glove on the right hand.
Okay.
So they use special chiral reagents molecules that are themselves handed, like Terashima's reagent or the CBS reagent. These help create the desired mirror image form of intermediate molecules.
But it sounds like getting it perfectly pure was still tough.
Oh, yeah. Achieving 100% purity of just one mirror image, that was a major, major challenge.
It really emphasizes how precise this chemistry has to be. Were there any totally different ways to build that core morphine structure?
Absolutely. Hudlicky's group came up with a really elegant one-pot reaction.
One pot. For something that complex.
I know, right? Imagine mixing a few simple ingredients in one flask, and through a cascade of reactions, bam, you build a big chunk of the morphine skeleton.
That sounds incredibly efficient.
It was. It allowed them to generate 3 of the key rings simultaneously, and then they used a very selective reduction step afterwards. Really clever stuff.
Wow. The source also mentions their synthesis of codeine, specifically. Codeine, anything noteworthy there?
Yes, definitely. They started with this molecule called diol 98, and they found something interesting about where reactions happened on it. That's called regioselectivity.
Meaning it reacts in one place but not another.
Exactly. When they tried attaching a silicon group circulation, it only went onto one specific oxygen atom. But when they used a different reaction, the Mitsunobu reaction, suddenly a different oxygen atom reacted instead, one at the C14 position.
Huh. So just changing the reaction changed where it happened.
Significantly. It really shows how subtle changes in conditions can completely redirect how a molecule behaves chemically.
It really drives home that morphine, even though it looks relatively small on paper, is a massive challenge, like a proving ground for synthetic chemistry.
That's a perfect way to put it. Successfully synthesizing morphine isn't just about getting the product, it's a benchmark, a demonstration of how sophisticated the field has become.
Okay, so that's the synthesis challenge. Now let's shift gears. Once the structure was known, the focus moved towards finding drugs without the addiction risk, right? The holy grail.
Exactly. That became the major goal after the structure was pinned down mid-century. And the source talks about how these new drug candidates started getting classified.
Right. Can you break down those categories for us?
Sure. So realizing the side effects of traditional opioids like morphine were a big problem, researchers looked for compounds that hit the opioid receptors in more specific ways. This led to roughly 4 main groups.
Okay.
Peripheral antagonists, delta antagonists, and kappa agonists. Think of them as different strategies for tweaking the system.
Peripheral antagonists. The name suggests they work outside the brain.
You got it. Drugs like methylnaltrexone bromide and alvimopan, they're designed to block opioid receptors mainly in the gut.
Ah, to deal with things like constipation from painkillers.
Exactly. Nausea, vomiting, constipation, those common GI side effects. But critically, they don't block the pain relief happening in the brain and spinal cord.
So you get the pain relief without the gut misery. Clever. Okay, what about delta opioid agonists? What's the idea there?
Delta receptors are another type of opioid receptor. Agonists activate them. And in lab studies, activating delta receptors showed promise for pain relief.
Any examples?
1067 from a company called Touré is one. It has a pretty distinct chemical structure. And interestingly, some delta agonists seem to have different side effect profiles than morphine, maybe better tolerated.
Okay, agonist activate. So delta antagonists must block them.
Yeah.
What's the point of blocking delta receptors?
Good question. Delta antagonists like BNTX, NTI, and TRK-851 have been looked at for various things. The source actually details how they design new versions of NTI with extra fused rings built into the structure.
Why add more rings?
The goal was partly to make them even better at blocking delta receptors, but also, surprisingly, to boost their antitussive effects.
Antitussive, you mean cough suppression? By blocking a delta receptor. That seems unexpected.
It is a bit, isn't it? But it highlights how these receptors are involved in more than just pain. TRK-850 came out of this work potent delta blocker, and it showed significant cough suppression in animals.
Sounds promising. Was there a catch?
There was metabolic instability. The body broke it down too quickly.
So back to the chemistry lab.
Exactly. They had to tweak the molecule, make parts of it less prone to being chewed up by enzymes. That led to TRK-851.
The improved version.
Right. It showed strong selective delta blocking, it was metabolized more slowly, and it had high oral antitussive activity in rats. The ED50 for cough inhibition, the dose needed to cut coughing by half, was quite low. Very potent.
That's a potential non-opioid in the
Right.
traditional sense. Cough medicine. Interesting. Okay, 4th category. Kappa opioid agonists. What's the story there?
Kappa agonists, like the early one U50488H and its derivatives, were explored as painkillers too. The hope was they wouldn't be addictive like Mo agonists such as morphine.
But there's always a but, isn't there?
Usually. The problem was side effects like dysphoria, feeling uneasy or unhappy, and even cyclotomimetic effects, things that mess with perception and mood.
Ah, so maybe less addiction potential, but other unpleasant effects. Not ideal.
Not ideal at all. So researchers started designing new kappa agonists with different chemical structures, non-peptide ones. Some designs tried to keep a specific part, the tyrosine moiety, found in natural opioid peptides. The idea was maybe that would reduce the side effects.
Did they figure out which parts of the molecule were key for kappa activity?
Yeah, through structure-activity relationship studies, or SAR. Basically, you make small changes to the molecule and see how it affects binding and activation.
Systematically tweaking it.
Right. And they found this address site region was really important for getting selectivity for the kappa receptor. That knowledge helped guide design.
And did that lead to better compounds?
It did. TRK-820 emerged from this. Highly potent, highly selective kappa agonist. In animal pain tests, it blew morphine and U-50,488H out of the water in terms of potency.
Wow.
And they confirmed it was working via kappa receptors using a blocker, NOR-BNI. Crucially, it seemed to have much lower psychotomimetic effects than morphine.
That sounds like a breakthrough. What happened in clinical trials?
Well, it was first looked at for post-operative pain.
Yeah.
And it did show pain relief in early human trials.
Oh.
But the safety margin, the difference between the effective dose and a dose causing side effects, wasn't considered quite wide enough for that specific use.
Okay. So end of the story.
Not quite. In a fascinating twist, it found a different niche. Doctors realized it might help with uremic pruritus.
Uremic pruritus. That's severe itching in kidney dialysis patients, right?
Exactly. A really debilitating condition. And there was thinking that maybe the mu opioid system contributes to itching, while the kappa system doesn't.
So blocking mu or activating kappa might help itch.
That was the hypothesis. So TRK-820, now called nalfurafine hydrochloride, went into clinical trials for uremic pruritus in Europe and Japan.
Did it work?
It did. Multiple studies showed significant reductions in itching scores, patients rating their own itch compared to placebo at both low and high doses.
Amazing. A drug designed for pain finds success treating severe itching. That really shows you can't always predict where research will lead.
It's a fantastic example of that.
Okay, let's move on. The source mentions 14-alkoxymorphinists. What are these?
These are basically morphine derivatives where you have an alkoxy group that's an oxygen attached to some carbon chain at the 14 position of the morphine skeleton.
And what's special about them?
They show a really diverse range of activities. Depending on the exact structure, they can be agonists, activating receptors, or antagonists, blocking them. Scientists have found good ways to make them, including related structures called indolo and benzopyromorphinins. They're really useful tools for studying the different opioid receptors.
The source also mentions clinically used drugs, like oxymorphone and oxycodone, which are morphine and 6-1s. How do these 14-alkoxy compounds relate?
Right. Oxycodone and oxymorphone are important painkillers. Researchers took the 14-alkoxy idea and combined it with modifications at the 6 position, specifically attaching amino acids there.
Why attach an amino acid?
The goal was to make it harder for the drug to get into the brain across the blood-brain barrier.
Trying to keep the effects peripheral again.
Exactly. Keep the opioid activity potentially for pain relief outside the CNS, but reduce central side effects like drowsiness or respiratory depression. And studies showed these modified compounds still bound strongly and acted as agonists at the mu opioid receptor.
Another strategy for separating pain relief from CNS side effects. Seems like a recurring theme.
It's a major goal in the field. And these 14-alkoxy compounds are also relevant for the delta receptor. Some of them, particularly the indolo and benzopyromorphinins, turned out to be even more potent delta antagonists than standard tools like NTI or NTB. So very useful for research.
It's amazing how changing just 1 or 2 spots on the molecule can flip its activity or target. What about the 14 amino derivatives, swapping the alkoxy for an amino group?
Right. 14-amino 4,4,5 epoxymorphinins. Here, the research aimed to improve on existing drugs like 14-hydroxycodinone and oxycodone by putting an amino group, nitrogen-containing, at that 14 position.
How did they make them?
One route involved nitrating the bane, another natural opioid alkaloid, and then converting the nitro group to an aminoid group. Then they made various derivatives by adding alkyl or acyl groups onto that nitrogen.
And what do they find pharmacologically? Any standouts?
Some showed potent antagonist activity blocking opioid receptors, but others, particularly a group called cinnamolamino-morphinones, were highly potent agonists.
How potent?
We're talking hundreds of times more potent than morphine in lab assays and much lower effective doses than morphine in animal pain models. Really powerful stuff.
Wow. Any specific examples of interesting activity?
Yeah, they made further modifications, like adding a chlorine atom at a specific spot on some 14 cinnamolamino compounds. One of these, MC-CAM, showed really long-lasting antagonist activity against morphine in animals.
Long-lasting antagonist.
Mm.
But it was derived from potent agonists. That's complex.
It is, and the blocking effect was delayed, which suggested it wasn't simply breaking down into another active compound, C-CAM. The mechanism might be more complicated.
So these molecules could be potent agonists and have delayed antagonist effects.
Some seem to. TAMO was another one mentioned. It acted like a mu agonist initially, but also showed a delayed ability to block morphine or fentanyl. Very interesting pharmacology.
It really underscores how fine-tuned these interactions are.
Yeah.
Okay, let's switch structures completely. Non-peptidic delta opioid ligands. What does non-peptidic mean here?
It means their structure isn't based on chains of amino acids like natural peptide opioids. And crucially, their structure isn't like the classic morphine scaffold either.
So wholly different chemical shapes.
Exactly. Discoveries like BW373U86 and SNC80 were important because they showed you could get delta opioid activity from molecules like diphenylpiperazines, which look nothing like morphine.
And did these activate or block the delta receptor?
Structure activity studies on SNC80 and related compounds showed they could be selective delta agonists, activating delta more than mu. Other non-peptidic agonist types included dirylocanals and 4-dirylaminopiperidines.
What about antagonists in this series? Blocking delta?
Those were harder to find, actually. DPI-2505 was one example from the diryl-methylpiperazine group. It's interesting because making changes to the nitrogen substituent, something that often creates more antagonists in morphine-like drugs, actually led to delta agonists in this non-peptidic series.
Huh. So the rules are different for different structural types.
Definitely. Shows that the receptors have distinct requirements.
And making these non-peptidic molecules, any special chemistry needed?
Often, yes. Synthesizing compounds like SNC80 required starting materials that were enantiopure, meaning only one of the two mirror image forms was present. Getting that often involves older techniques called classical resolution to separate the mirror images.
Okay, now for something really different. Salvinorin A. This one's fascinating. A kappa agonist with no nitrogen.
That's the headline, yeah. Salvinorin A is a natural product, a neoclaridyne ditropine. Complex structure, unique carbon skeleton, and it hits the kappa opioid receptor despite lacking that basic nitrogen atom usually considered essential for opioid activity.
So it breaks the typical rules. What are its effects?
It's known as a potent, non-serotonergic hallucinogen. Its unique structure and pharmacology make it a huge target for synthesis and for understanding kappa receptor function.
Have chemists managed to synthesize it from scratch?
Oh, yes. Given its complexity, 7 chiral senders that deterpene-core several top groups like Evans, Burns, and Forsyth Hagawara have reported total syntheses. A real challenge.
And have they modified the natural molecule too?
Yeah.
To see which parts matter?
Yes, semi-synthesis, starting with the natural product and tweaking it. That's given a lot of insight.
So what's crucial for its kappa activity?
Generally, changes at the C2 and C3 positions aren't tolerated while activity drops, but adding an ester group at the C3 hydroxyl often keeps or even boosts activity.
Interesting. What else?
Modifying or even removing the C17 lactone ring seems okay. The double bond between C8 and C17 is tolerated. Removing the furan ring hurts binding, but reducing it is okay. And flipping the 3D arrangement at C12 really tanks the affinity.
Wow, a very specific set of requirements for this unusual molecule. Okay, let's look at another strategy. Making new structures from existing opioid drugs like naltrexone. And naltrexone blocks receptors, right? So why modify it?
Right. Naltrexone is mainly a mu antagonist used for addiction treatment. The idea here was to use its existing complex structure as a starting point, but then perform chemical reactions that dramatically change the core skeleton, hoping to create entirely new molecules with potentially useful and different opioid receptor interactions.
So using it as a scaffold for something new.
Exactly. They tried making things like orthoesters with an oxybicyclo 2.2.2 octane skeleton, though those turned out to be unstable.
Instability. Always a problem. Any successes?
Yes. An unexpected reaction created a different ring system, an oxybicyclo 2.2.1 heptane. Even more dramatic, a reaction using ACECl actually broke the C16N17 bond in naltrexone, creating a totally novel basic skeleton.
Breaking a bond in the core structure. How did that happen?
The proposed mechanism involves a temporary intermediate, an oxazolidinium ion, and suggests that the 3D arrangement of electrons favored breaking that specific bond over others. Pretty sophisticated chemistry.
And these completely new structures, did they still do anything at opioid receptors?
Some did, remarkably. Another reaction sequence led to a molecule that was missing the entire D ring of the original morphine structure, but it still showed strong binding to the mu receptor.
Missing a whole ring, but still binding. That's surprising.
It really is. It shows that drastic changes are possible. They also made complex things like propellants and even trimers, 3 units linked together, starting from naltrexone derivatives. One trimer showed potent pain relief in mice.
Incredible chemical creativity there. Okay, nearing the end, the source talks about twin and triplet drugs. What's the concept?
This is about designing single molecules that contain 2 twin or even 3 triplet pharmacophores.
Pharmacophores meaning the active parts of a drug.
Exactly, the parts that interact with the biological target. The idea is that one molecule could bind to multiple receptor sites simultaneously.
Why would you want to do that?
It could lead to increased potency, better selectivity, or maybe even novel effects by hitting different targets or different sites on the same target. They're also called bivalent ligands, chimeric ligands, hybrid ligands. Lots of names.
How are they built? Are the active parts just strung together?
Sometimes. You can have conjugate drugs where the pharmacophores are linked by a chemical spacer. Or they can be fused directly or with a very short linker. Or even merged where the active parts are blended into a shared structure.
What kind of combinations have they made?
Lots of opioid combinations, varying the spacer length to see how it affects binding. Also, combinations targeting different opioid receptors like delta and kappa together, which can be used to study how these receptors might pair up heterodimerization.
And triplet drugs are just more the same concept.
Basically, yes. 3 pharmacophores linked together. They can be symmetrical, all 3 the same, or non-symmetrical, all 3 different, potentially offering very complex pharmacology from one molecule.
Sounds powerful, but are there downsides to making these bigger molecules?
Definitely. Things like drug likeness, basic properties needed to be a successful drug, and getting across the blood-brain barrier become big challenges. These larger molecules might not be absorbed well or reach the brain easily.
So a trade-off between complexity and practicality.
Often, yes. Though the source notes that twin drugs with longer spacers might genuinely bridge 2 binding sites, while the mechanism for shorter linked ones is less clear but still potentially useful for design.
Okay, last section. 3D pharmacophore identification for K-opioid agonists. What's a pharmacophore model in this context?
It's about figuring out the essential 3D arrangement of chemical features a molecule needs to bind and activate the kappa opioid receptor specifically.
Why kappa specifically?
Because, as we discussed, selective kappa agonists are potential painkillers without the more side effects like addiction or respiratory depression. We know several classes of molecules hit kappa receptors, but we don't have a perfect picture, like an X-ray structure, of the receptor itself.
So how do you figure out the 3D requirements without seeing the receptor directly?
You use computational methods, homology modeling, where you build a model of the kappa receptor based on known structures of similar receptors, and ligand docking, where you computationally try to fit known kappa agonists into that model.
So build a virtual receptor, then see how known drugs fit inside.
Exactly. By looking at how different known kappa agonists, peptides, morphines, arylacetamides, salvinorin A, etc., can overlap in 3D space within that model, you identify the common essential features, specific spots that need to be hydrophobic, or places that need to accept a hydrogen bond.
And that collection of essential 3D features is the pharmacophore model.
Precisely. It's like a 3D map or blueprint showing what a molecule needs to look like spatially to be a kappa agonist.
What did the model described in the source reveal?
It identified specific hydrophobic areas and hydrogen bond acceptor points that lined up with different parts of known kappa agonists. It helped visualize how these molecules likely orient themselves when they bind. They could even classify the known agonists into different types based on how they fit the model.
So it's a tool to understand binding and, I guess, to design new potential kappa drugs.
Exactly. You can use the model to screen new molecules virtually or to guide chemists in designing structures that have a higher probability of hitting the kappa receptor effectively. It's a key part of modern drug discovery.
Wow. Okay, this has been a really deep dive covering so much ground in opioid chemistry, from just figuring out how to make morphine
Right, that initial huge challenge.
to these incredibly sophisticated strategies for designing targeted drugs, multi-part molecules, using computers to predict activity. It's clear the field is constantly pushing forward.
Absolutely. The complexity of the opioid system itself is immense, but the chemical ingenuity being applied is just remarkable. Fine-tuning existing structures, creating entirely new ones, aiming for that perfect balance of efficacy and safety.
Developing selective agonists, antagonists, these twin drugs.
Mm-hmm.
The toolbox is getting incredibly diverse.
It really is, and it holds so much potential, not just for pain, but as we saw with nalfurafine, for other conditions too.
Which really leaves you wondering, with this accelerating understanding of the receptors and these creative chemical solutions, what are the next big breakthroughs going to be? Better pain relief, sure, but what else?
And maybe even more intriguing, what completely unexpected applications might pop up for these molecules as we learn even more about how they work in the body? It's a fascinating space to watch.